Hello, everyone, and thanks for joining our webinar. Our webinar is entitled Spotlight 59, an ultrasensitive sequencing workflow for liquid biopsy samples. The presenters today are going to be myself, Christian Ionescu Zanetti. I'm the CTO and one of the co-founders at Fluxion Biosciences. And we also have Drew McCusick, who's a application scientist at Swift Biosciences, our collaborators on this project.
Hello, everyone.
So thanks again for attending. It looks like everybody has joined up, and we will get started. Before we get into the material, I wanted to give you a quick overview of what we'll be talking about today. First of all, this workflow is primarily geared towards liquid biopsy samples. So we'll talk a little bit about the promise and the challenge of this specific sample type. A lot of workers in the field have tried to address these challenges using molecular barcoding or single molecule unique identifiers. We're going to talk a little bit about the progress there. After that, we will get into the Accel Amplicon technology from Swift Biosciences. This is the targeted amplification technology that is being used as part of this ultra-low, ultra-sensitive sequencing approach. And we're also going to talk about a few applications of that technology to liquid biopsy samples. The body of the presentation will be regarding the Spotlight 59, which is both the panel and the workflow that works in conjunction with EraseSeq, a computational pipeline that allows us to call very low frequency variants. And as part of that, we'll present a few sets of analytical validation data that demonstrate the sensitivity and specificity of this approach. Finally, we'll show a couple of pilot studies looking at the validation of this workflow for IsoFlux liquid biopsy workflows. Specifically, IsoFlux is a circulating tumor cell-based way of interrogating cancer patients in order to determine the somatic mutations that they present from blood. And we'll present here data from clinical samples that were obtained as part of that effort, and show the full sample to workflow, sample to answer rather workflow for that particular platform. All right, so first, the promise of liquid biopsy. This is a field that has seen a lot of advances in recent years, and it really offers a lot of promise in terms of being able to non-invasively look at the genetic makeup of various human cancers. This is important in a couple of different areas. One, of course, is early detection and diagnosis. A lot of the most deadly cancer types, like lung cancer and pancreatic cancer, are detected late in the disease cycle. Most of the patients are stage 3 and stage 4, and that really impacts survivability in a negative way. And so a number of efforts are underway, including companies like Grail, for detecting the presence of tumor cells early by using blood markers, and this is a promising way of doing it. Other studies, the majority of the studies actually have so far have looked at patients that are far along the disease cycle and have been usually diagnosed for quite a while and are undergoing various treatments, and oftentimes have failed the first round of treatment. And so these types of cancers are advanced, and the physicians are typically looking for targeted approaches. So they've already gone through typically chemotherapy, the traditional first line treatment, and then are going into second and third line treatments. And for those treatments, it's very important to determine the presence or absence of mutations, because that often gates what what types of treatments will be efficacious for those patients. So those are the two different areas where this is being applied. There are a number of good publications on this. This is a recent one from Science Translational Medicine, where a number of these patients, these are advanced patients, but about 75% of them had detectable cancer mutations from cell-free DNA in the blood. Of course, the certain tumor types had lower levels of detection, and most importantly, the frequency of these mutations goes way down when the stage is stage 1 or 2. Not sure if you can read this graph here, but basically between stage 1 and stage 2 patients, only about 50% of them had any detectable cell-free DNA. And that's why it's very important for us to increase the sensitivity and specificity of these types of assays. So besides having a really low mutant allelic frequency in this assay, in this sample type rather, another challenge here is the fact that the cell-free DNA input is quite low. So you've got a sample number of copy issue that you have to deal with. So some samples have as much as 30 nanograms, which is roughly 9,000 copies, but more often you are in a range of 10 or even 1 nanograms being extracted from each sample of cell-free DNA. And so if you're looking at alterations that are present either at 1% or below, you have very few mutant copies. And one of the reasons that we have developed this method with Swift, and one of the great things about the Swift Amplicon panels, is that they have very low starting material requirements, low input requirements, and we have data to show that 1% mutant allele detection is easily obtained from as low as 10 nanograms input. And you'll see why this is important, both in general and for this workflow in particular. So you've seen a little bit about the promise of this cell-free DNA approach. The challenge, of course, is the signal to noise, which is the ratio of signal to noise in these sample types, especially at early stages in the disease cycle, is very low. There are often variants that are present below 1%, where 1% is the ratio of the mutant to the wild type allele. So that's true across the board, both for cell-free DNA and for circulating tumor cell samples. It's been shown by a large number of publications that there's a significant fraction of alleles present below 1%, and as you saw in the previous slide, the copy numbers are also quite limiting. So people have taken a few different approaches to address this. There are commercially available tests developed in a CLIA environment where people have developed their own kind of bioinformatic pipelines. Here we're going to focus on things that are commercially available to all labs and all cancer centers, things like molecular barcoding that is provided as a panel, or methods that have been developed and published on previously. So unique molecular identifiers or molecular barcodes have been recently developed, and they've been shown to significantly reduce the errors that are present in each read. And the way that's achieved is by having a consensus read for each molecule, and you'll see in a sec how that's done. Now, they do have limitations. One of them is they require a very high read depth because each molecule needs a number of reads of the same sequence to be, collapsed into one consensus read. And importantly, there's a limited amount of the starting molecules that can be barcoded. And so if you look at the literature, the state of the art of the percent molecules integrated with barcodes is about 20%. There are many approaches, like duplex barcoding, for example, where you have two different barcodes, where that percentage is only about 2%. And so you need a lot of starting material to make these work. The other issue with these approaches is that despite the fact that they reduce the per read errors a lot, they don't work quite as well for actual variant calling down to really low percentages. As you'll see in the next slide, they actually have been shown to only be sensitive to about 0.5% in commercial panels, and the published data really doesn't go much below 1%. I think that's because some of these limitations that I've just talked about. Now, when we started these efforts at Fluxion, we were compelled to look at this as a possible method to do the low variant allele frequency calling to use barcoding methods. And one of the main reasons that we did not pursue this method is because of the high copy number input requirements. And as you can imagine, for circulating tumor cells, we have a low number of inputs, as few as 500 copies. But it turns out that even for ample inputs, there are some real limitations when it comes to calls, to making calls by using molecular barcodes. So these are two recent publications. One is from Hyatt et al., one is from Peng et al. One of them deals with a technology from QIAGEN that has been recently commercialized. And basically, what they show is that they describe, first of all, the way that these molecular barcodes work, and by incorporating a tag and then replicating that tag, so you can then you can then assemble these reads into families, each corresponding to one starting molecule. That results in very accurate reads for that one molecule. However, the overall false discovery rate or false positive rate and the overall sensitivity is just not that much better than just using regular amplicon sequencing. So as an example, in this paper, they quote a false discovery rate of 20% and a sensitivity of 94% for higher variants, but only 4% for variants in the range of 0.2 to 0.4%. That's Hyatt et al. And this is really the region that we were interested in. Peng et al. Has similar results. They have really good sensitivity for variants that are 1 to 2%, so above 1%, with 3 false positives. When you go below 1%, again, the sensitivity drops below 60%, and the false positive rate goes up. So just the thing to remember is that these are really promising approaches, and they work really well in terms of reducing per read error rates, but they still have limitations in their current form as far as variant calling per se. So with that, I'm going to pause here for a second and pass things on to Drew, and he's going to be talking to you about the Aston Amplicon technology and Swift's work in this area of liquid biopsy. Thank you.
Thanks, Christian. So as Christian explained, the Excel Amplicon product line is Swift Bioscience's solution for generating multiplex PCR targeted libraries for next generation sequencing. You can see the workflow here illustrated on the left. It's quite a simple and fast workflow. In just about 2 hours, you can go from your starting DNA template input material through to a sequenceable library, as shown here with dual indexing for the Illumina platform. It's 2 steps. In the first step, there's a multiplex PCR where you incubate your DNA with primers specific for your loci of interest. generate your targeted amplicons. And then in the second step, the indexing step, there is an adapter ligation that occurs to convert those amplicons into library molecules. It's a simple single tube per replicate workflow. So this is very helpful for the Array-seq Spotlight 59 panel that we will be talking about shortly. Because if you're generating multiple replicates for this workflow, then having just a single incubation for each one of those is quite helpful. It's also a single kit. You don't have to purchase separate kits for the primer pool versus the library generation reagents. So that also simplifies the workflow. This technology also allows for overlapping or tiling amplicons over regions where, for example, with whole exon or whole gene coverage, where your targets of interest may be larger than can be covered by a single amplicon. So rather than having to split these targets into multiple tubes, the technology allows for tiling these amplicons simply with the bioinformatic requirement that the primers indicated in blue here, which are inside of those tiled regions, must be trimmed. And that's something, again, that the Array-seq workflow also takes care of. So not something that you'll have to do on your own. As well as your main panel, included in the Spotlight 59 panel and our other panels, we have the ability to spike in sample ID targets. And so these are approximately 100 amplicons total, which target exonic SNPs with high minor allele frequency, as well as a handful of gender identification targets. And what these allow you to do is, without taking too many of your reads, without taking up too much of your flow cell, it allows you to both verify whether multiple samples are derived from correct individuals. For example, in longitudinal studies where you may have a baseline sample and want to doubly confirm that the sample that you're following up with is from that same individual, as well as if, for example, you're looking at multiple samples from the same individual, such as liquid biopsy plasma versus normal or tumor FFPE samples. And so these amplicons are spiked into your main panel, as illustrated here, in a low molar ratio compared to the main panel. And this allows you to do germline variant detection for these sample identification fingerprinting amplicons at a low level, and then preserve your high-depth sequencing for somatic mutation detection, for example, in cancer sequencing. So another feature that's important to discuss, especially when we're reaching low allele frequency variants and trying to detect low allele frequency variants in liquid biopsy samples, is coverage uniformity. You can imagine that it's very important that all of your targets are amplified and detected at a similar depth of sequencing, regardless of how large that panel is. So the definition that's been adopted widely in the field of amplicon sequencing is to analyze what percentage of your targets are covered at 20% of your mean depth of coverage or greater. So for example, if you can imagine sequencing your oncology panel, in this case, the Excel Amplicon 56G oncology panel, to an average depth of 5,000x coverage, you're asking yourself the question, what percentage of my amplicons that are in that panel are covered at 1,000x or 20% of that mean or more? And so what you can see, that's the 20% threshold is this dark purple bar. And what you can see is with high molecular weight genomic DNA, in fact, the 56G panel achieves effectively 100% coverage uniformity, meaning all of those amplicons are sequenced at that 20% or higher threshold. Where this falls off with a lot of other technologies is that if the amplicons aren't designed properly for liquid biopsy templates like cfDNA, circulating cell-free DNA, that takes a hit to the coverage uniformity. And this is because if you have a target, for example, that requires primers to be spaced 200 bases apart from each other, cell-free DNA's main peak size is 165 base pairs. So that amplicon would not be effectively produced, and you would have a hit to your coverage uniformity. Again, with the 56G panel, the Spotlight 59 panel, and all the Excel amplicon panels, we strive to make these designs such that, in fact, that coverage uniformity is maintained above 90 or 95% throughout the panel. And so overlapping amplicons and coverage uniformity are important features of the panel and how it functions. At the end of the day, most of us are interested in variant calling and detecting low frequency variants, especially from liquid biopsy samples. So the analytical validation that we have performed with the 56G oncology panel was using the Horizon Diagnostics quantitative multiplex reference standard, which is HD701 catalog number from Horizon Diagnostics. This sample contains each of these variants at the allele frequency specified here, where this is the reference base in the reference sequence, and this is the alternate base, which is known to be present at this frequency in the sample. And over 10 replicates with the 56G oncology panel, as you can see here, we were able to detect with 100% sensitivity each of these variants at the expected allele frequency down to 1% allele frequency. And so this is where from 10 nanograms of this DNA, the Excel amplicon products are currently supported. Similarly, on another control that's available from Horizon Diagnostics, the 1% multiplex cfDNA reference standard. This is a standard which has a size profile that is similar to cell-free DNA, and you can see to the point of, again, coverage uniformity, allowing deep enough sequencing for each of these variants to be detected down to 1% frequency. We did also have the opportunity to participate in a study with some translational research samples, and these are 8 different blood samples isolated from individuals with varying stages of oncology presentation. We took these samples, the cell-free DNA was isolated from these samples using a QIAGEN kit, and 10 nanograms from each sample was placed into the 56G oncology panel. This is a profile of each of these individuals with their different clinical presentations, mostly ovarian, some pancreatic, and colorectal cancer. And what we were able to see when looking at both the cell-free DNA and the matched normal adjacent and FFPE tumor DNA was that in some of these cases with sort of earlier stages of cancer, in fact, some of these variants were detectable in the FFPE tumor DNA, but in neither the normal adjacent tissue nor the cell-free DNA. Whereas in some of these samples, in fact, the same variant which was detected in the FFPE tumor was also detectable, again, down into the 1 to 5% allele frequency range in the cell-free DNA. But then to the point of specificity, these same variants were not detected in the normal adjacent tissue. And so to get to the Spotlight 59 panel, which is the highlight of this webinar, these are the specifications for the panel that was co-developed with Swift Biosciences and Flexion Biosciences. And what we have here are the specifications for the panel, the input amount of 20 to 100 nanograms per sample, which allows you to perform the replicate analysis required. It's flexible to a variety of sample types, so each of these circulating tumor cells and circulating cell-free DNA being liquid biopsy samples, as well as solid tumor FFPE samples. As I mentioned, the workflow and other specifications are very similar to our other Excel amplicon panels. This particular panel has 277 amplicons covering oncology targets in 59 genes, as well as the sample ID spike-in panel that I mentioned. The average size of the amplicons, again, is compatible with cell-free DNA templates. It's an average size of 140 base pairs. And then our on-target percentage and coverage uniformity are above 90% for this panel. As Christian will be describing to you shortly, with the EraseSeq bioinformatics workflow, this technology is capable of reliably detecting allele frequencies below the 1% threshold. And importantly, that pipeline is also a HIPAA-compliant cloud-based analysis platform, which is included with the product. And so at this point, I'll also just briefly show the targets that are included in this panel. There are 59 genes. This is a list of those genes and the number of amplicons which we have in the panel covering each of those genes. As I mentioned before, with the concept of the overlapping contiguous coverage, there's a number of these genes which have that, and they're indicated in light blue, as well as in the dark blue here, TP53, we actually have full comprehensive coding exon coverage for that gene. It's about 40% of the panel has tiling regions in it. So that's sort of an overview of the XL Amplicon technology and the Spotlight 59 panel. And at this point, I will be happy to turn it back over to Christian for discussing the rest of the workflow.
Okay, thank you, Drew. So after this, we're going to go in more detail about Spotlight 59, and a lot of this will be talking about the analytical performance metrics in terms of variant calling below 1%, and then we'll have some data from clinical samples as well. Before we go into the details of the computational pipeline, I just wanted to briefly go through kind of an overview of the workflow, just so you know where all of the elements fit into this. So first of all, we assume that the customer starts with DNA prep from either CTC, cfDNA, or FFP samples that are interesting in terms of clonality. Followed by that is a target amplification step using the Spotlight 59 panel. So that's really what the panel does, is enable very low noise target amplification that's compatible with all of these different sample types, and that's what Drew's been explaining in detail. The sequencing is done to for the lowest detection limits. We require 20,000x depth of sequencing. For slightly higher limits of detection, we can use data that's at 8,000x, and that's per sample. As you'll see, this includes replicate samples, so this depth is per starting sample, and that includes multiple replicates, as you'll see. As far as the analysis, we have a computational pipeline that's very robust, and it was implemented in a cloud-based environment. We've made sure that all of the data transfers are encrypted and the storage is HIPAA compliant, and at the end one gets a report with the variants that were detected. I'm going to spend most of this talk explaining EraseSeq. Erase stands for Elimination of Recurrent Artifacts and Stochastic Errors. It's really the brainchild of Nicholas Campos-Hughes, who's heads up our bioinformatics here at Fluxion. So you've probably seen a lot of images like this before. I just wanted to go through briefly the types of errors that you see in these kinds of samples, and these are really the errors that are responsible for the background that we see in most sequencing runs. And roughly speaking, there are two different classes of errors. There's a type that we call recurrent artifacts, and those really appear because of regions of the targeted panel where you just have a higher chance of getting an error during replication. Now, replication can either be the targeted amplification step, or it can be happening later in the process during the sequencing. the actual sequencing run. So as you know, you start from a template. When you amplify a certain region, you can get errors. Most of those errors are random. Some of those errors happen to occur with a higher probability. So when that happens, you'll get something that looks like a real variant when you're talking about these low allelic frequencies. And that's what's indicated as a red cross here. There's another type of error which is called a stochastic error or jackpot error. And this just has to do with where these errors occur in the amplification cycle. So as you might imagine, if you start with just a few hundred copies and you have an error happen in one of the first few sets of amplifications, that error is going to be really prevalent. The error can happen early on, and then it's going to get propagated through all the way to the end product, and that again is going to look like a relatively high, relatively prevalent variant in the sample. So what ends up happening is you've got a noise floor in these sequencing runs that typically runs from about 0.01% to about 1% frequency. And what we did here is we basically used a standard caller and we relaxed a lot of the call quality metrics. What this allows you to do is, of course, it allows you to call a lot of the low allelic frequency true variants that are present in these samples, but it also allows a lot of other calls. And this is a representation of what this looks like for a particular sample. So now the challenge here, of course, is figuring out which ones of these are real and which ones of these are not. So here's just a set of real calls in the range of 0.5 to 1.5%. This is part of the sample set I just showed you earlier. We're just focusing in on a few of the calls. And because we prepared these samples by spiking in tumor cell line DNA into a healthy background, we know that we basically know the answer for these analytical runs. So we know that some of these are real. Some of these variants are what we would call true positives, and others fall into one of the two categories I just presented earlier. They could be recurring artifacts or they could be stochastic errors. So we tried to develop a method to eliminate both of these classes of errors. Now, what Nick suggested is that we sequence the sample multiple times. This is not something that people haven't thought of before in terms of replicating a measurement. As a biophysicist, I understand that replicating the measurements is something that you want to do, especially when you have a lot of noise, and that accurate determinations of a measurable quantity are dependent on that. There was a lot of resistance, however, to taking this approach, and a lot of the objections to it that were raised internally were because of the difficulties with creating the right workflow and doing this in a cost-effective and time-effective manner. So what we've really done since this original idea was put on the table is figure out a way to do this in a way that's readily implementable, and a lot of this had to do with working with Swift and adopting their panels. And I'll tell you why in a minute. But first, back to the errors. So as you'll see, these are the calls that were high in S1, the first sample, and this is the result from a sequencing run starting with that sample material. S2 and S3 are subsequent replicates of that workflow. And these are the calls at the same loci with the same base change. Now, just by looking at these three replicates, it should become obvious that some of these calls should not be included in the call list and are probably errors. So I told you about these types of early replication errors. And clearly in sample 1, something happened here that was different from samples 2 and 3. These calls can be eliminated by basically comparing the 3 samples. Now there are other calls that are present in all the samples, but some of those are what we call recurrent artifacts. So the way that we eliminate those calls in our bioinformatics pipeline is by comparing to a very detailed background model that we created by running a large number of non-tumor derived reference samples. So one of the things that we have at our disposal, because we have a known panel with a known chemistry, is this background model that was created by Swift and Fluxion, and that is included in the pipeline. So this is something that we had to do to bring this up, but that doesn't have to do every time a customer runs a sample. So basically what the pipeline does is it applies a statistical test between a number of replicates that are all served by the Spotlight 59 panel and the background model that we've built up over a large number of healthy samples. What this allows us to do is eliminate both classes of errors and get to a set of true positives for each set of sample files that's submitted. So how do we do this? So we basically, one of the things that people have previously done is to create some kind of very simple consensus, meaning instead of having a single run with a threshold and calling above that threshold, people have said, well, we'll do multiple runs, we'll call above the same threshold, and we'll just look at a union of those calls. And in these noisy samples, that's actually not what you want to do, because that is very limiting in terms of telling being able to tell the difference between a real call and the error background at that position. So what we implemented is a statistical test and call it confidence score that differentiates places where the sample, the target sample is different from the reference background. And this is a plot of the same positions. It lines up to the same plot where I had the allelic frequency, plotting our log of confidence score versus position. Now, what I'm also plotting here is, instead of plotting everything as a small dark dot, you've got these red dots, which are the real variance. And as you look at this threshold here, this is a threshold in the confidence score. You might be tempted to say, well, look, I can understand that you're picking out the real variance. Some of them are really obvious. For the ones that are low down, close to the noise, it's actually a very difficult task to reliably detect those and reliably separate them from the background. What you're not seeing is that because this is a log scale. Actually, if you look in a linear scale graph, you'll see that these are very well differentiated from the background. So this is the same plot that you just saw with those two variants that are closest to our limit of detection, but now they're plotted on a linear scale. So all that to say is we have quite a bit of room here to differentiate true variants from false positives. Now, if you go back to the original plot, where instead of confidence score, we're plotting allelic frequency versus position for detected variance, it's clear that we're doing much better than an approach where one just has a threshold. And we're detecting variants that are real, even when they're look to be down in the weeds as far as a allele frequency-based background. So finally, I want to talk about some of the analytical tests. Here we did what all of the other papers that I've referred and people typically do here, which is to look at 4 different cell lines. We started with 4 tumor cell lines that are listed here. We mixed all of these at 1% and mixed that into normal DNA and came up with an analytical test figure, mixture rather, and then sequenced that and put it through our pipeline. Additionally, we have an additional sample I'll talk about where we did 1/4 of this, so it's 0.25%. So what this results in is a sample that has a number of variants at ranging between 0.2 and 1%. And these are the cell line sequencing results on the same panel. This is the expected frequency based on the spike percentage and the variants present and whether they're present in 1 or more cell lines. And here I show the detected frequencies and mutant read numbers that were obtained using Spotlight 59. And Spotlight 59 basically uses 4 different replicates, and it puts together the read numbers from all of those replicates, and it uses a statistical model to figure out which of these calls are true positives. So as you'll see in this case and in other cases, we're able to detect all of the true positives without the false positive background. Now what this really allows you to do is make the most of your sequencing depth. What I mean by that is if you were to take the same sample on the same panel and put it through a standard sequencing workflow, and then have a caller that had relaxed enough quality metrics so they could call things down to 0.5%, which is what we did here, you would get a good number of these calls, so the sensitivity is pretty good down to 0.5, but you would also have a significant number of false positives. In this case, the false positives are about equal in number to the true positives that are detected. And this is just for a range of 0.2 to 1%, where most of the variants are closer to 1%. If you do this below 0.5%, obviously your results are going to be significantly worse. So what I wanted to do is just give you an idea of basically the previous graph by looking at different regions where variants are located. Okay, and here we use a standard variant caller, term low freq, and we set the parameters in such a way that you could still detect a significant fraction of calls between 0.5 and 1%. And if you do that, you'll see that there are 10 true positives, and 10 true positives are detected in the 1 to 2%. As Drew said, these panels work really well above 1%. And if you were to tighten those quality metrics, you could probably, by losing some of the sensitivity down low, get really good sensitivity specificity above 1%. That's our starting point. As you drop below 1%, your sort of true positive and false positive rates start to be closer to one another, and you start losing, both sensitivity and specificity. Now to the right, again, is the output of our pipeline at a similar sequencing depth to the single run. And you can see that, we're able to eliminate all of the false positives that were present and retain the true positives where, most of them are in the range of 0.5 to 1% in this case, but there are definitely variants in the range of 0.2 to 0.5%. As I alluded to earlier, we also did experiments that are in a range that is basically 1/4 lower than the experiment I just showed. And this really gets to a lot of the range where I think both the cell-free DNA and the CDC samples end up being in terms of true mutant variants. So here we have variants all the way down to the range of 0.05 to 0.3%. I think the detected allele frequency for the lowest variant is 0.07, but you really are probing the range of 0.3 to 1% here. So what you can see is the the performance is not surprising given what we've just seen. So at 1 to 2%, the panel does quite well using standard calling technology. But that performance drops off below 1% where you have really 2x the number of false positive calls compared to the true positive calls. And in this range, it's very hard to use this for de novo detection of variants. Again, the Array-seq caller performs really, really well for all of these ranges, and it's really detecting all of the variants that we expect in those ranges, in this case without false positives. And I just want to point out, we really had no cutoff. Because of the way that the model works, you don't need to set an allelic frequency cutoff. That's really based on the noise level at that particular position. As we saw, most of the targeted region has really low noise levels, far below 0.1%. Okay, so after showing you some of the analytical data, I just wanted to compare that to what's been previously published on using both standard callers and molecular barcoding approaches. And you have to bear with me. This is a pretty dense slide. So Foundation Medicine has done a lot of great work in terms of sequencing and profiling solid tumors, and where they really set their threshold is the 2 to 5% range, where they have a really high sensitivity of 98% with very low false positives. And again, just to look at apples to apples, I've set the false positives reported to be per 10 kb of panel sequenced. Now the 56G panels from Swift are really low noise, so they perform really well in the 1 to 5% range with perfect sensitivity in the samples that were tested. Now I want to talk a little bit about molecular barcoding technologies. Some of these are commercially available. Guardant Health is a test, so it's not something that one can purchase, but it's something that one can order. And what they report in Landman et al. Is that they can detect down to 0.3% allelic frequency with 85% sensitivity without really reporting an analytical false positive rate. They also have a look at a range of 2 to 5%, and they have, I think, perfect sensitivity specificity, or close to perfect sensitivity specificity in that range. Now, the couple of molecular barcoding papers, and one of them is related to a commercial barcoding panel, molecular barcoding panel from QIAGEN. They go down to a 1% allelic frequency with 90% sensitivity, which is again really good, but I think not orders of magnitude above what one can do with really low noise panels in terms of targeted sequencing. Where things really break down is in the range below 0.5%, which is why I have plotted this here. Again, this lines up to the bar graphs I showed earlier, but if you go in and really try to assess these below 0.5%, you're not going to get very good performance. For Spotlight 59, the performance we've tested a lot around is the 0.2 to 0.5% range. As you saw in the experiments that we did, we had 100% sensitivity with no false positives. The same applies, of course, to 0.5 to 1%. And I've got an asterisk here because we, haven't done 99 experiments. So, but we have a relatively large number of variants in some of these ranges. So for example, in the below 0.5 range, 0.2 to 0.5 range in the second experiment, we had 20 variants. So that sensitivity is expected to be greater than 95% over repeated experiments there. So finally, I wanted to talk about one way to use this clinically, and really it has to do with what I told you we started with at Fluxion is we have a circulating tumor cell isolation instrument, that does really well in terms of sensitivity to tumor cells and in terms of the purity of tumor cells. And we've put a number of those samples through this workflow. So the way that this works, just briefly, we have sample collection, which is whole blood and EDTA tubes. It's processed within 36 hours, and our instrument does the removal of all of the red blood cells and most of the white blood cells. And what you end up with is a sample that is 5 to 15% tumor cells, but really low copy number. And what happens is that even for those samples, because of tumor heterogeneity, you often get into the range of sub 1% in terms of mutant alleles. So we used, this is ongoing work where we used our NGS kit, and then we put it through the Spotlight 59 workflow that I've been talking about. That includes the target amplification, the replicate data uploads, and then the computational outputs. So this is a sampling of the results we've obtained for clinical samples. These are all diagnosed cancer patients. We did focus on types of cancers like pancreatic cancer that are typically hard to assay using circulating tumor cells, and we've focused on getting a good mix of earlier stage tumors as well as later stage tumors. About half of those are stage 1 and stage 2. As we expected, this performed much better compared to our previous sequencing efforts in that we were able to detect alterations in a majority of these samples. We were not able to detect alterations in healthy controls that were put through the same protocol. We have also done a number of just quality control experiments where we do things like biological replicates and comparing the calls from, let's say, 2 different samples that were drawn from the same patient on the same day, and those showed very high concordance. We have concordance to primary studies that are ongoing, and that data is not here, but this is something that's obviously really important, and we're working on that. So I just want to conclude here, and then maybe we'll get to some questions. I hope I've shown you that Spotlight 59 can achieve very accurate detection down to 0.1% with very low noise. This is really what makes this feasible, is really the Swift Amplicon panel technology. And that's for a couple of reasons. One is because it's a single tube Amplicon technology, and that means for workflows like this where you require replicates, you can just have a 4-tube reaction, and so that's feasible. And because this panel is tailor built to this workflow, it ends up being affordable to the end user. It has all of the required reagents included through the sequencing runs. Again, what this also relies on is a computational pipeline, and that's provided as a cloud-based application free of charge. And I believe that this is one of the first workflows to really enable calling a variance down to below 0.5% from these either cell-free or cell-based liquid biopsy samples. So again, what's in the box, just so that people understand what Spotlight 59 entails, is the targeted amplification for all of the samples required, the sample tracking ID, which is what Drew talked about. It's really an important thing to have for just quality control and tracking of sample source. And all of the library preparation reagents are also included at this time for Illumina sequencers only. And as I mentioned, the RACE-seq bioinformatics solution is part of the package as well. All right, with that, I'm going to end, and thank you for your attention, and I'll try to address some of the questions. All right. All right, so one of the questions was regarding overlapping amplicons. I believe we've already answered that, but yes, there are overlapping amplicons, and Drew, correct me if I'm wrong.
That's correct, yes.
There's another question, how are amplicon duplicate reads identified without fragment barcodes?
So currently, being an amplicon panel, there will be multiple amplification events from each template sequence. So this workflow currently does not deduplicate based on those amplification events. But as Christian has talked about, the workflow still allows the low allele frequency detection through the use of these replicates.
That's right. So we do not, just to be clear, we do not use any molecular barcodes or unique molecular identifiers as part of the workflow. We just found that for this level of performance, it wasn't necessary to do that. We found that the replicate measurements of noise background as well as sample were sufficient for detecting down to 0.1. Okay, so in the circulating tumor cell application or workflow, there's a question that says, when you mention CTC plus background, what is the expected CTC ratio? So for our NGS kit, we typically have about 500 cells, and of which 5 to 100 cells are expected to be tumor cells. We end up at about 5% or more tumor content. However, because of the tumor heterogeneity, we end up often with variants that are at or below 1%. There's also a question about custom panels. Yes, it's possible to create custom panels. I think because you know how this strategy works, one would have to not just create a custom panel, but also then work on creating the bioinformatic, basically error model for that particular panel. But it's definitely possible to use custom panels. There's a question about the false positive rate of 0.25 per 10 kb on the FM panel. This is probably, this refers to our table that I've shown earlier. All of these were calculated based on what was in the specific publication that was referenced. I think I'll have to look at that offline, but I imagine the 0.25 per 10 kb means that there was, say, a 40 kb panel that had one false positive in the specific range. There's also a question about how confidence scores were calculated. That requires a more in-depth conversation, and it's also part of the secret sauce for the RACE-seq pipeline. But I think I'm going to have to take that privately. It's basically, what I can tell you is it's a statistically robust comparison of the expected noise and the actual sample readings at that location. There's another question regarding the DNA input requirements. It reads, 4 multiplex PCR reactions at what input DNA mass? So again, that's all in the specifications for the Spotlight 59 panel. We specify, I think, 20 to 100 nanograms input. These experiments were done at 40 nanogram input, so that's 10 nanograms per replicate. So that's ideal. We believe that this works down to 5 nanograms per replicate input. So that's a total of 20 per sample. There's another question. The attendee asks, how many total reads were obtained per specimen, not per reaction? So where you saw the table earlier, let's see if I can go back here. Where you saw the table earlier, these numbers of reads listed here were numbers of reads per specimen. So these are sums of the numbers of reads over 4 reactions. All right, there's another question about custom panels. Again, it's possible to use this approach on custom panels, and I believe Swift offers custom panel creation. The only additional work that has to be done is experiments to determine the backgrounds for that custom panel.
Yes, that's correct. And we would normally engage those requests on a case-by-case basis. So you can contact us, and we'll start with the target lists and go from there.
There's another question that asks, when is the molecular barcode added to the Spotlight 59 panel? And again, let me just be clear, no molecular barcodes are used here. In fact, we assessed the use of molecular barcodes. We decided it was not the best approach for this sample type. So there are the only barcodes that are used to identify the different sample replicates that go into sequencing. There's another question regarding how many samples can be run on one flow cell of the MiSeq. I believe for the V3 chemistry, the answer is 6 samples can be run.
Yeah, and it comes down to just taking the total number of reads for that flow cell and then dividing it by the size of the panel, so that the 277 amplicons and the depth that you need. And so exactly, if you do 5 or 6 samples on a MiSeq V3, that's dividing 25 to 30 million reads over those 5 or 6 samples, and that's going to give you that 20,000x coverage that's required. So that's either, ideally in this workflow, that's 4 of those 5 samples, so 4 replicates of each of 5, to give you that combined 20,000x coverage.
Okay, thanks, Drew. There's another question. How does the error model change from lab to lab, and what does the lab need to validate a specific error model? The error model is lab independent. We have run these analytical samples at different locations and on different sequencers. I will say that it most definitely is dependent on the type of sequencer used and the sequencing chemistry. We have done these runs on the MiSeq, the MiniSeq, and the HiSeq. If you're using a different technology, we would probably have to very carefully look at the background models again and do an analytical validation. But so it's not there's no lab to lab variability per se, but there is a validation required for different sequencing technologies. All right, and just since we're approaching the hour, I'm gonna just take a couple more questions, and then if I don't get to your question, we have your email, and I'll attempt to address those directly by email. Another question is it recommended to run samples in duplicates to run through EraseSeq? So again, there's a workflow that includes the sample replication and includes the replicate runs. We use 4 replicates in this workflow. There's a question about cost per sample. I can't speak to exact costs just because the costs are slightly different in different geographies, but you should expect the cost to be on par with other low frequency detection panels that are available out there, like the unique molecular barcodes. So I can address that privately if I know where you're located. But there's another question about samples run in duplicates run through same barcodes or different barcodes. As far as the sequencing is concerned, each replicate is treated as a different sample. So you've got 4 sample barcodes, 4 sequencer sample barcodes per starting specimen. There's another question about the coverage required. The question is, 20,000x coverage does not take into account the uniformity, so a depth of 20,000x is not true for all amplicons, ? How to calculate if one wants this per amplicon? Just to be clear, the 20,000x depth required was a mean depth, and we do have uniformity requirements, but the uniformity requirements are just basically that you hit the spec uniformity of the amplicon panel that is used, in this case the Swift amplicon panels. There's another question regarding the amount of cell-free DNA available. The attendee asks, taking cell-free DNA into account, sometimes there's not enough DNA to run replicates. And that is true for some samples. So we believe a 20 nanogram input would work well. If your starting sample is below that, as we often have for the circulating tumor cell samples, where we only have a few hundred copies, what one has to do is do some other preamplification. In our case, we use whole genome amplification, and I can go into more detail. If your sample has been whole genome amplified before it goes into the Spotlight 59 panel, there's a slight degradation in performance, but we still, roughly you should think about the performance we get at 0.1% now being more like 0.3% allelic frequency for confident detection of variants. Okay, I think I'm going to end the public session here. I will address some of the questions that have come in privately, and I thank you all for attending. It's been a pleasure telling you about our work, and if you have any other questions, feel free to contact us via the Fluxion website. And the product is basically launched now. We will have another launch presentation at the Association for Molecular Pathology in conjunction with Swift at their corporate workshop. So thanks for attending. Have a good rest of your day.
Thank you, everyone. Goodbye.